Hi everyone, it's Dr. LeBlanc with Unit 14B, HFR strains and genetic mapping. The learning goal for this lecture is to understand the use of HFR strains in mapping genes using conjugation and partial gene transfer. HFR strains were discovered in the 1950s by Luca Cavalli-Sforza. He discovered a strain of E. coli that was very efficient at transferring chromosomal genes, and he called them HFR for high frequency of recombination. So we have our F plus cell. We know what an F plus cell is. An F plus cell is an E. coli cell with its own chromosome plus an additional F factor. An HFR cell, on the other hand, is a E. coli bacterium that has the F plasmid, F factor, F episome integrated into the E. coli chromosome. Integration of the F factor into the chromosome occurs by recombination. The origin of transfer, which is right here, is now part of the bacterial chromosome instead of being located on its separate F episome. When an F factor integrates into the chromosome, it creates an HFR cell. Okay, what happens when the F episome excises or comes out of the host chromosome? Well, normally it comes out as one piece and just remakes an F episome. But what happens if it comes out imprecisely? Then what's called an F prime factor is created. So we have our HFR cell with an integrated F episome. If it loops out from the bacterial chromosome and carries with it some of the bacterial genes, it creates an F prime factor. So an F prime factor is an F episome that's carrying some bacterial chromosomal genes with it. When that happens, it usually leaves behind a little piece of the F episome in the host chromosome. And the F prime factor carries a portion. In this case, it's carrying two genes, a gene for proline and a LAC plus gene. Okay, what happens when you have conjugation between an HFR strain and an F- strain? An F- strain, remember, has no F factor at all, and an HFR cell is one with the F episome integrated into the bacterial chromosome. The origin of transfer now is part of the bacterial chromosome. So when it transfers, it may end up taking some of the host chromosome with it. When you start to transfer the chromosome, you can transfer some of the bacterial genes along with it. So if you have a cell, an F- cell that is pro minus and LAC minus, and an HFR cell that's pro plus and LAC plus, you can end up transferring that genetic information and converting this cell now into a LAC plus pro plus. So we have our HFR donor cell, which retains its original genotype, and our F- recipient cell now that has some additional pieces of DNA in it. If you let the transfer go for a short time, you can transfer one of those genes, the LAC plus gene here. If you let it go for a longer period of time, you can end up transferring both of those genes. The F- cell received a short segment of the HFR chromosome and has become LAC plus but remains pro minus. If you let the transfer go for a little longer, then the F- cell receives longer segments of the HFR chromosome and it becomes LAC plus and pro plus. What is an interrupted mating? Well, an interrupted mating means that you let the transfer go for a short period of time or a specific length of time and then stop the transfer at different intervals. These types of experiments were used to map the order of genes along the E. coli chromosome. The chromosome of the donor strain is an Hfr strain, just like we just saw, and during mating, part of the chromosome is transferred in a linear manner to the recipient strain. The order of the genes along the chromosome can be deduced by determining the time various genes take to enter the recipient cell. Woolman and Jacob used two E. coli strains. The donor strain, which was plus, but it was sensitive to streptomycin, and a recipient strain that was minus, but it was resistant to streptomycin. So if you allow the conjugation to occur and you plate the bacteria on Petri dishes that contain this antibiotic, only the recipient cells will grow. The donor cells will not be able to grow because they are not resistant, they are sensitive to streptomycin, and streptomycin will kill all the donor cells. Okay, have that in your head? All right, here's the experiment. You start out with a flask of bacteria and you'll allow conjugation to occur. At different periods of time, you separate these two cells from each other by putting them in a blender and separating the two cells. But turning the blender on will separate these two cells. They won't be able to withstand that agitation. Okay, you plate the cells then on solid growth medium with streptomycin, and then you can plate them from this one plate onto a whole bunch of different plates that determine their genotype with respect to azide and whether or not it can grow with T1 phage, whether or not it can make its own lactose or galactose. So if you do those tests after 5 minutes, what you determine is although that the cells are streptomycin resistant, none of these genes have been transferred. These cells are not threonine or leucine positive. They're not azide resistant, or they can't promote growth of bacteriophage. They can't make their own lactose and galactose. Okay, no genes were transferred after 5 minutes. What happens after 10 minutes? Well, after 10 minutes, now all of the recipient cells are able to produce their own threonine and leucine. And some of them are azide resistant, and a couple of them can grow bacteriophage, but none of them are LAC or GAL+. Okay, at 15 minutes, no more changes occur. You just get more of what you've already transferred. After 20 minutes, you see some LAC+ cells, and after 25 minutes, you just start to see some GAL+ cells. So from these data, Woolman and Jacob constructed the following genetic maps. The order of the genes transferred was, after refining it even further than what that data that was shown, they were able to determine that the order of the genes on the E. coli chromosome was threonine, leucine, azide, TON, LAC, and GAL. They also identified various Hfr strains in which the F factor had been integrated at different places in the chromosome. By comparing the order of the genes among all these different strains, they were able to first of all demonstrate that the E. coli chromosome is circular and that this was the order of the genes. And when we refer to the position of the genes along this map, we refer to it in minutes. So at an arbitrary place for starting with 0, all of these genes are at different minutes apart on the E. coli chromosome. This is the type of data that you would obtain for their interrupted mating experiment. This is the duration of mating in minutes along the x-axis, and along the y-axis, you have the percentage of F- recipient cells that have received the gene during conjugation. The most important aspect of this particular type of graph is the time in between you see the first appearance of cells with that particular genotype or phenotype. The distance between the genes is determined by comparing their times of entry. Here is the time of entry of the LAC-Z gene, and this is the time of entry of the Gal-E gene during an interrupted mating experiment. Therefore, these two particular genes, Gal-E and LAC-Z, are 9 minutes apart along the E. coli chromosome. The rest of this up here doesn't really matter that much. What matters most is the initial time of entry when these genes first appear in the recipient cells. So in summary, bacterial strains with an F episome integrated into their genomes are called the term Hfr for high frequency of recombination. When an F episome imprecisely excises and carries a piece of the bacterial chromosome with it, it is called an F' episome. When conjugation occurs between an Hfr strain and an F- cell, no episome, part of the host genome can be transferred into the F- cell. Interrupted mating experiments between Hfr and F- strains can be used to map the order and distance between genes in the bacterial genome.